Welcome to Genetics 4, subtitling this the Punnett Square. Reginald Punnett, Punnett, is the person credited with making these squares, and I'm just going to bring one out, enlarge it, not going to tell you what the letters are, but basically just tell you a little introduction. You remember up here on the top, gametes, male gametes, are the sperm. And what each of these letters identify is the possible genotype of the gametes. Now you got to remember this is 1N, right? The sperm are 1N, and then over here on the left, the female gametes are also 1N. And then when we combine them, which we will, that's really like thinking fertilization occurred, and then we're going to restore the 2N genetic state, and then we can show the genotype in these squares. And then a lot of times, especially for beginners, it's neat to put the phenotype underneath here. So anyway, it's a two-dimensional grid, kind of shows you how the gametes get together, and one assumption is that each gamete has an equal chance of meeting each of the other gametes of the other sex. And every once in a while I run into something comical, and I just thought this was a great little picture. You can see that, I guess we'll do, this is the sire, this is the dam, and then this is the progeny, right? And this gentleman's got horizontal lines. The female has vertical lines, and then the progeny has the cross. It's got both the and I guess, what, that would be like a roan cow where both alleles are expressed. So that actually, now that I think about that, this is codominance. Wow. Didn't know that, or didn't have that plan to say that, but kind of comical. So, I'm just going to be naming certain things about the Punnett square. If you haven't heard of what the Monohybrid Cross, that's basically when you're only looking at one locus. And let me bring you a couple examples here. Now, this happens to be from the plant world, and I guess maybe the reason I'm including it is because, plants follow the same genetic principles, but I always like to put, if this was an animal, I'd like to put the sire up on top, the two alleles from the one locus, right? Remember? So this would be the capital R allele, that it's in one sperm. The other sperm has the little r. And then over here, I like to put the female on the left side. And so I'll say it's a female. So then it's capital R, little r. And then you combine them. So like this would be what the zygote would look like genotypically. And then actually they've got the phenotype in a graphic. So large R. And did they say that large R is dominant? Must be, because you only need one copy of a large R to get the smooth seeded offspring. So then only when there's two little R's do you get the, what do they call it, wrinkled seed, okay, offspring. So look at this. The genotype is, there's one of those, two of these, and one. So you'd have a 1 to 2 to 1 ratio. That's the genotypic ratio. RR, and then there's 2 big R, little r, and then 1 RR. So 1 to 2 to 1 genotypic ratio, but the phenotypic ratio is 3 to 1 since it's a dominant recessive, so 3 to 1. So be aware of that terminology. Here's another example, and I'm picking out some examples that follow my convention of putting the sire up here on the top portion of the grid and then the dam, the mother, on the left side. They have black up here, but it's actually, obviously, it's capital B and then little b and then capital B, little b. So then you can see again, we have a 1 to 2 to 1 genotypic ratio, but a 3-to-1 phenotypic ratio. 3 black, they might, I don't know if they're talking about dogs or whatever, it doesn't really matter what they're talking about, to 1 brown offspring. So phenotypically 3-to-1, genotypically 1-to-1. Okay, so now we've got a dihybrid cross, which is looking at 2 loci. Loci is the plural form of locus. I wouldn't say 2 locus, I would say 2 loci. And then I just want to show you this. I don't know what animal we're talking about, what traits really. It looks like color of something, but I don't know if they're talking about whatever. It doesn't really matter. I want to point out how you've got 4 possible genotypes of the sperm, let's say, and 4 possible genotypes of the eggs, and then you combine them. So it's 4 by 4. So you're going to get 16 possible combinations. And then, of course, these then would be the zygotes. They don't have underneath here what the phenotype is, and that would be really pretty good if it did, because then you can kind of see the genotype of the offspring and the phenotype of the offspring. Now, I just wanted to show this. It's a screenshot of some program I saw. It's basically the Punnett Square calculator. It's a professional one, and I'm not going to tell you all about this, but anyways, it tells you the probability of the different genotypes, and if you mentally add that up, that's 100. It looks like they had an 8 by 8 square. I'm looking up here in the upper right corner here, and so there's 64 possible squares. And when you're writing that out by hand, that's kind of long, and if you know the principle, then you don't have to go through that long hand grid work. You can have a computer program, or what they call a Punnett Square calculator. Now, I wanted to use the roan cow as an example, so maybe what I should say, or it's actually a shorthorn. The breed is shorthorn. The color is roan. And if you're looking at that far right there, a roan, and we had this in an earlier genetic lesson, and sometimes they use capital W for white, but you get a combination of white and red. And for shorthorn cattle, roan means there's separate hairs of red, separate hairs of white. It's not like half the hair shaft is red and half is white or whatever. It's just like pure red or pure white. So the cows could be all red, all white, or if it's the heterozygote state, then you're going to see this animal. So this animal's heterozygote. And I'll just get him out of the way because I just want to build this a step at a time. So I'm going to stop, do something, and then come back. So I hope you're saying, oh yeah, this is a monohybrid cross. We're just looking at 1 locus. We happen to have 2 alleles possible. Remember, you could have 5 alleles. But when you're talking about an individual sire, they would only have 2 possible alleles, and then the dam only 2 possible. So remember, this would represent the genotype of the eggs. They're either large R or little r in my example. And then this is the genotype of the sperm, large R, little r. And then I combined them. So we got large R, large R, large R, little r. I always put the large R first, then the little r, and then, so in our genotypes, remember, is on top, okay? And I'm going to put the phenotypes underneath here so you can see that. But obviously we have a 1 to 2 to 1 genotypic ratio, right? But hopefully you remember, and what we'll have is that same, and this is interesting because then this animal up here is red, pure red. This animal is pure white. Now some have capital W in some places when they talk about shorthorn cattle, but some places have little r. So white, red, and then 2 roan. So we have a 1 to 2 to 1 phenotypic ratio also, because this is a codominant trait. You can go back. So now I'm going to pause this thing and I'm going to just write in what I just said as far as the phenotype of these animals. Okay, so now the phenotypes have magically appeared. And of course, large R is going to give us red. Then we have two roans, and then we have a white. So since there is codominance here, the phenotypic ratio equals the genotypic ratio, right? The genotypic ratio, again, is 1 to 2 to 1. And that's what our phenotype is, the coat color. We have 1 to 2 to 1. Now, if it happened to be, if large R was a dominant trait and overrode the little r, then we'd have the 3 to 1 phenotypic ratio. So hopefully you can understand this. Monohybrid cross, very interesting Punnett square. I've had many students watch me do these on the board and think they can do them, and then the test comes along and they founder or flounder, whatever you want to call it, because unless you do it and talk through it, especially you get a dihybrid cross, maybe you've got to have a 4x4 square and know how the alleles interact. Easy to watch, but it takes practice. Finally, here's the list of those beautiful illustrations I used. See you next time.